thank you all for coming. My name is Steve McDowell. I'm serving as the interim dean of the graduate school this year. I want to welcome you to our special presentation. I'll just make a few introductory comments about Professor Sorin Istrail. He's now the Julie Nguyen Brown Professor of Computational and Mathematical Sciences and Professor of Computer Science at Brown University. And he's the former director of the Center for Computational Molecular Biology at Brown University. He's also has experience both in the private sector and government labs. He was the senior director and then head of informatics research at Solera Genomics, where his group played a central role in the construction of the sequence of the human genome. And this paper was published in Science in 2001, which is one of the most cited scientific papers to date, with about 20,000 citations. Before that, he was also, he founded and led the Computational Biology Project at Sandia National Laboratories from 1992 to 2000. So he's got a tremendous background in government labs, in the private sector, as well as in academic research. We're really happy to have him here, and this project has been a collaboration between the graduate school. Dr. Brian Barton is here. He and I worked with Dr. Don Carr from FSU Health, and Dr. Carr is both the director of the Pepper Center here, but has also been working with the Office of Research, or the VP of Research, to try and bring together FSU Health across the campus too. So I'm not sure, Dr. Carr, if you want to say anything more, but yeah. So she's both organizer, but also kind of a host here, because this is the center is in this building here. So with that, we'll welcome our guest, Dr. Sorin Istrail, and thank you all for coming, and look forward to a presentation and some discussion.
Streaming? Yeah, streaming. Where camera? And looks like the program is placed down. So it is up. It is up again, and it's going to
Okay.
yeah, because yes, it goes
So
up,
it
it goes south.
Okay. Do we know what?
Going to bring another dongle from my office, and that one works.
Sorry.
No. Oh yeah, no. Listen, take that out.
We have a thumb drive here.
Yeah, there you go.
Let's use this. This is the most convenient solution.
Yeah,
This is the less problem.
yeah.
So it's double click. There we go.
Nothing is working. Nothing is working today.
But that's it's a long file. Okay.
It's winning. It's bottom.
Okay. Copy naman. It sounds alright. And then I'll start sharing the screens. Everyone is just. Okay. So PDF is that right?
Sorry.
That's the. Sure. Yeah, that'd be another way to.
So it's visit. No.
Okay. Welcome to sharing screen. And just a moment, please. So make sure I have everything.
How do you?
That's what was.
Right, thank you very much. Thank you, Steve, and thank you, Don, and the university for inviting me, and thank you, my son Lee Istrail, who is a resident of Tallahassee, and we are so delighted to be here with him and his wife, Rebecca. And it appears that some music collaboration between Lee and Steve led to computational biology talks. So I am honored and delighted to be here. All right, so I felt that in preparation for this talk to pick some topics that I worked on related to the interest of the university in pursuing a mission statement and a vision for the new health project. So I thought I should talk about some of my results related to computational biology that can lead to medical applications. And I gave a grandiose title of 3 topics: haplotypes, gene regulatory networks, and protein folding. They are all areas of my research over the years. And when I thought about putting them all together, I got PowerPoints for all 3. It was very hard to start moving around. So I'm going to talk about one of them. And in this talk, maybe I'll mention a little bit. So I'm going to talk about gene regulatory networks, because this in particular is a topic that benefits from causality, this 21st century intellectual challenge, unresolved for 1,000 years, that is now getting much more research and progress, and very important in applications to medicine, algorithms for genomics that are applied to medical bioinformatics. So this is work related with my collaborator and professor, Eric Davidson from California Institute of Technology, who passed away in 2015, unfortunately, who was
my most important biology collaborator. We worked 15 years together, and I'm going to talk a little bit about the great problems that he proposed. He was uncontested the leader of molecular biologists in the area of gene regulatory networks and gene regulatory systems. So I'm going to talk about this beautiful set of problems. As causality is a theme that was continuously pursued by scientists, but continues to be a mystery, and after 1,000 years, we still don't know what the cause is. Recently, computer science was able to make advances in causality, and it's very interesting that Davidson had his own concepts of causality, and I'm going to call this part Davidson causality. And you are going to see that mathematics plays a fantastically important role in understanding the cell, which is an object full of liquids. Everything in the cell is liquids. How can we get from that to computer science to mathematics, and how to be able to do causality inference in this cell? So that's why I'm saying mathematics, it's almost unreasonable effective in the regulatory genome, in the parts of the genome involving gene regulation. So I have an overview of some of the topics I'm going to talk about, and we'll start with the gene regulatory networks, the gene regulatory network that Davidson and collaborators studied and published many papers about it. And it's all about sea urchins. The model organism that Davidson lab studied was the sea urchin. So we'll hear a lot about sea urchins in this. And we'll talk about the sea urchin genome, the sea urchin transcriptomes, all the genes that are expressed, the proteins. We're going to talk about something called the logicome. The logic, the Boolean logic in the genome, the regulatory genome of the sea urchin group. And then we'll have a parallel between the electronic computer and the regulatory genome, which is a fascinating comparison between the two. The cell is such a complex object that needs an information processing system in the cell. How is the information system in the cell organized? It's fascinating. You have only liquids. How do you do this? And then I'm going to show how Davidson actually axiomatized his definition of causality. And we will complete the presentation with this seminal work by Davidson and Peter of a Boolean model that shows causality completeness for a Boolean, for a gene regulatory network of 50 genes. Causality completeness is the only result in the literature in any sciences that find all the causes. So the causality completeness of a gene regulatory network, which is a wet lab system, experimental system, and the causality was proved by computational biology methods, which is a fascinating result. So we published a number of papers together. I'm very proud of them. I published in 2019, a few years after his passing, a survey of all the problems that I worked with him and the big question that he asked. And I call it Eric Davidson Regulatory Genome for Computer Science, Causality, Logic, and Proof Principles of the CIS Regulatory Code. And I can say that we did many things together. One of the most important paper is my postdoctoral work with him logic functions of the genomic cis-regulatory code. How do you find these Boolean logic functions in the cell? And we did many things, including I was part with him in the science publication of the genome of the sea urchin, and also another article in the same science, the transcriptome of the sea urchin, and many other interesting things. So I'm going to ask two questions that Eric asked, just to show how to ask really big questions. I have to say that I learned from my father how to ask big questions. He was one of those people that liked impossible problems to solve. And in my journey, I tried to recognize those big thinkers in the collaborator that I have. So I have quite a few, Eric Davidson, and Craig Venter. So here is the first question that Eric Davidson asked. So in the literature, there are papers published of various genes and their regulatory regions from the gene that is explored by cause and effect, by inference of the causality of all the connections in the regulatory region. And those papers were published in journals. And he asked the question, how many such papers exist that talk about the regulatory region that is analyzed with the ultimate level of rigor, which we call the Davidson criterion, which is loss of function in vivo and mutagenesis of the DNA and loss of function in vivo. So you want to really see that the perturbation removes the biological function. So he asked this question, find all the papers involved. So this was a really big challenge for computer scientists because some of the papers were published well before the PDFs as a structure existed. So how can you extract with software from the literature the entire universe of ultimately validated cis-regulatory architectures? So this was indeed a big problem that one of my PhD students, Ryan Tarpine, in his PhD thesis, solved it and found out that the universe, the entire universe, is probably about 800 such ultimately validated cis-regulatory architectures. The second problem, which is the problem that Peters and Davidson solved with their Boolean model, can be formulated as follows. And look at the ambition of this question, the grandiosity of the demand. So the question is, can a formal computational model of gene regulatory network be developed that is completely predictable of its experimental output? We're talking about computationally predicting the experimentally measured output in the lab. So in the groundbreaking work of Isabel Peter and Eric Davidson, they developed this Boolean computational model called GRN network equation that is almost completely predictive of the experimental output. The extraordinary scale of this achievement applies to the sea urchin embryo gene regulatory network and is sufficient to predictably explain almost all spatial regulatory transactions underlying a large portion of the embryo of the sea urchin in very early stages, pre-grass to embryonic process. They also published a book that appeared in the last year of him being alive, Genomic Control Processes, Development and Evolution. And that presents in full detail this pioneering solution of the sea urchin embryo via this almost completely predictable biological model. We'll see very soon the gene regulatory network. It has about 50 genes So the fundamental question is, do they have all the genes, or there are other genes they didn't discover yet that are still part of this process? And they prove no, they had all the genes, and the way they prove it is through computation, if you can imagine, which is a fascinating result in computational biology. So I'm going to say a few words about the world of sea urchin and how we get to Boolean logic functions in the sea urchin. So here is a picture of a genome from the Celera human genome paper, where I was one of the co-authors. And what you see here are chromosome by chromosome displayed, and where there are genes or regions of important biological function, they are annotated on the gene. You could see there are many regions which are empty, the so-called junk DNA, interregion regions, and so on. But overall, it's a extraordinarily beautiful picture to see the whole genome displayed with the state-of-the-art knowledge on it. And what you see here in the corner, you see an electronic computer, the hardware of it. So you are going to see there is a very clear parallel between the two, and some of the hardwired DNA patterns are what is happening in the hardware sense, as we will see soon. So the dogma of biology says that the DNA, which is represented by this twiggling yellow line, ribbon, whatever, is bent, and there are proteins that are binding to DNA patterns, and those proteins recruit other transcription factors that are floating in the cell, and they get together in such a way that protein machinery can be formed. So this is the DNA dogma of how the protein machineries are happening based on patterns and protein DNA interactions on the DNA. If you represent schematic the regulatory regions of various organisms, you are going to see there is a pattern in common. You know, every line is a regulatory region, and the squares and the circles are proteins that bind to it. You could see, you know, the sea urchin regulatory regions of two genes are up in the upper part. For mouse, you see a regulatory region which has very much similarity. A rat is another one, and here is the human. And the so-called cis regulatory code problem is to try to understand what is the code here. Those are hardwired pieces of DNA that incorporate a certain machinery. Here is the zebra fish, and other genes that are involved in their regulatory regions. I used to give talks about Eric Davidson's work, and his main claim to fame before the gene network was the endo-16 gene that he discovered, and together with 300 docs and postdocs and faculty visitors like myself in his lab. But for this one was only docs and postdocs, they discovered how this gene endo-16 works. So the gene is represented by this double line. The proteins are represented by square that bind to the double line. And this is the regulatory region of endo-16. It's a gene involving the gut of the embryo of the sea urchin. And through systematic perturbation, with 300 docs and postdocs, you can cut the DNA in so many places to find cause and effect, the inference of causality. They discover this workflow, this flowchart that is encoded in that piece of DNA, and they realize that there are, you know, similar with electronic circuitry, there are gates, there are activation arrows and inhibition flags, and many other things that are happening there. And this explain exactly how the gene operates based on the transcription factor that bind. Here is a pseudocode of the previous program represented in actually programming code. And you will see that some of the inputs depend on time. They are time dependent. They are liquids that are time dependent. And some of them are just constants, and some of them are just Boolean. Look at this, if then else. This is how this program works. So here is Eric when he was younger presenting his seminal theory. So here is the gene again at the top, endo16, and the pseudo program at the bottom, much easier to read. And if you look at how this program works, it goes from left to right, and various parts of it are like programming components that work to regulate the gene. And in the end, the top level of the program involves repression, a switch amplify, and here is the pseudocode, the most basic way to understand it. Now, here is the gene regulatory network, the one that is proven completeness by Isabel, Peter, and Eric Davidson. And here is our endo16 gene. It's a gene that goes outside the network. So every gene that is in a certain organ or cell type has a gene regulatory network that regulates it. Here is the entire 50 gene gene regulatory network, endomesoderm gene regulatory network. So things are so complex, we need to use electronic circuitry for displaying. I mean, we use the same software as for electronic circuitry. The understanding is each of these bar horizontals are genes. This is where the gene binds by showing this bent arrow. And each gene goes and operates as input to a regulatory region of other genes. Those are all transcription factors that work in the network. And this is the question: do we have all the genes? Do we have all the transcription factors? And imagine the complexity of such an operation to prove completeness. Only Eric could ask such questions, and they were able to do the job. And we're going to talk a little bit more about it. So those diagrams that I show they are called the view from the genome. In the genome, all the circuitry is overimposed on each other in a potential existence way. But when you are in a particular cell, you get the view from the nucleus, which means only some of the genes are available, and all the others are shaded because they exist there but are not active in the particular cell that we are. So we are in a nucleus of a cell. So this view from the genome, view from the nucleus is a paradigm which is very important. So my work with Davidson went through many aspects of genomics. The genome we published in Science, the transcriptome we published in Science, the logicome we published in Brazilian National Academy of Sciences, and the regulatory genome we published as a comparison between the information system called regulatory genome and the electronic computer. And we have this beautiful comparison that we did. So I'm going to say a little bit about some of those things. Here is me at Celera going to Caltech for periods of research. And Eric, the biology professor, said, everybody does experimental work. You go in the lab and do your part. So here I am, with my sweat of killing small sea urchins because I messed up some experimental thing. But anyway, it was a fascinating time. I was learning. Basically, Eric said, I cannot talk to you unless you do experimental work because you don't understand anything. You need to be at the level of the experiment. So it was so fascinating to listen to him and be like one of his students. So this our paper was the paper with hundreds of authors, and I was still at Celera, and my group did the first assembly of the sea urchin at 2x coverage. He was looking for his transcription factors, and it was a fascinating, privileged way to be involved and get credit for it. The transcriptome, we worked as a next project. I was one of the senior authors responsible for a third of the project. And this was the project of finding what are the genes expressed in the, so it was a new technology with chips that were able to discover patterns expressed as genes. And we cataloged the entire transcriptome of the sea urchin, and this was the paper in Science. The logicome. This was what my postdoctoral work with him was about, to find the logic functions of the genomic code. How can it be logic function, true and false, in the world of liquids? How can we model it? How can we discover principles? Because without them, we cannot do inference, we cannot understand what is causal and what is not. So in his work, he worked on the sea urchin genome, and then the Drosophila patterning of the embryo genome. You have the network, also a patterning network of 50 genes, or roughly 50 genes, like was the endomesoderm for the sea urchin. So those are the two model organisms that were leading our understanding of regulatory genomics. The Drosophila work was done at the Institute for Advanced Study by Mike Levine and his and Eric Davids at Caltech with the sea urchin. So I can tell you a little bit how Boolean logic shows in at the level of liquids in the cell. Here is the first one. It's called an AND logic, and it's from the patterning of the Drosophila, of the fruit fly. And you could see there is a very interesting way of different measurements that can be recorded in the truth table. And you could see that when the two of them are present, the signal is there. When one is not, it's exactly the Boolean truth table, and you get the AND operator. And many things like this happen. The second one is a short repression operator. The next one is signal-mediated toggle switch. Various operations very relevant to the life of the cell that can be translated into a truth table, and you get the logic functions of the genomic. Here is the looping. It's the dating operation. What I'm calling dating is like eHarmony. What's happening is that there are proteins that bind to DNA, and they stick to each other to bend the DNA together to enforce them to make proteins together. And it's like enforcing protein-protein interaction to make things happen in the cell. Fascinating things. This is the linker module and so on. So we catalog various types of operations. Some of them are logic, some of them are probabilistic. They used certain statistical mechanical equation due to Bolori-Davidson and so on. What's happening in the cell is the DNA is the green one, and then proteins start binding to it, and they start forming assemblies among proteins in the cell. Some of them are insulators to give them the chance to work in parallel. It's a fascinating world in the cell. It's like a Lego building construction. So here are classification of the different type of logic function that we found. Here is a more detailed naming of the operations that are functional in the cell and so on. And for each of them we have biological analysis in detail of the truth table of what's happening. So it's fascinating how all these measurements become classified as logic functions of a certain type. Here is the conclusion of our logic function paper. There is a common repertoire of cis-regulatory modules across the species. There are many interesting things. Occupation is causal. CRM outputs are intrinsically coordinated on site occupancies. And in all cases where there is a qualitatively unique factor site interaction, the consequences of a site mutation and the absence of occupancy due to the absence of the DNA factor are equal. So this is our famous equation. 0 cis is equal to 0 trans, which basically says you get the same disruption if you detain the DNA so the transcription factor cannot bind, or if you forbid the transcription factor altogether to be in the cell. 0 equals 0. So I'm going to move next to causality because it's Davidson axiomatized causality. So this is a paper that showed up exactly in the year in which he passed. I'm so delighted that he was the greatest writer. He published over 400 papers, and his eloquence in writing was a model for all of us. And he could write with this speed that in the last year of his life disappeared. And this is a phenomenal article about his thoughts on causality. So I encourage you to read those papers. So I'm going to just go quickly over his writing of epistemology and the causality inference, and he talks about Karl Popper. I know that Dawn has some interest in philosophy to her daughter. Here it is, a good source. So Karl Popper was, as we know, this enormous scientist who so Eric criticizes that developmental biologists throughout the 20th century suffered inescapably from Karl Popper criticism of inductive scientific process, that all it takes is a single instance to the contrary to prove an inductive mechanism idea wrong. Like the white sheep versus a black sheep. If you see only white sheep, you form a theory, but the first black sheep that you see is killing your theory. So it's very interesting at a philosophical level what is going on. But I'm not going to have time to go into the details. I'm going to go only through his axioms. Like everybody else, instead of defining causality, he axiomatized. If you take a book on causality nowadays, they start by saying, we don't define it, we cannot define it, but we are going to axiomatize. We're going to say some of the axioms causality satisfies. So we're talking about Eric Davidson axiomatization of causality in the domain of molecular developmental gene regulatory networks biology. Axiom 1, rooted causality explanation is anchored in the genome. So unless you link it to the genome, you don't have a rooted causality explanation. Unrooted causality explanation is phenomenology, stem collector, based on a small part of the system. And this is the fundamental thing that he desired. This is the goal, the thinking big man. Explanation of system-wide control mechanism for developmental process must begin with recognition of the elements of the regulatory sequence, but you want a system-wide explanation. You don't want just one gene affects another, and this is my paper published. I want a system-level explanation. All the genes involved in this interaction. Axiom 2. No sum of unrooted causality explanation equals a complete rooted explanation. So the solution of the mechanism, you want to find the mechanism of biology, is impossible if only a minor fraction of the components active in a process of the interactions is involved in the analysis. No additional fragmentary, unrooted explanation, even infinite many of them, will ever sum up to a complete genomically rooted explanation. The reason in the enormous combinatorial number of degrees of freedom with which the interactive control system of development can be, and the evolutionary have been assembled. So you want somehow to go through the system level explanations. Success, axiom 3, success measures must be assessed system-wide. The usefulness, success, failure, validation, or refutation of an explanation emerging from experimental system development biology must be assessed at the level of entire system. This test must challenge the ability of the explanation to predict the behavior of the system as a whole, or the behavior of a large sector of it that include many individual components and their interactions. So it's all about system level. Axiom 4, this is one of the most beautiful things, almost poetic. From the sea of phenomenology to sea of causality. Inverting the island in the sea metaphor. You see, everything is non-causal. We are in the ocean of non-causal things. Causality is in the very small islands that we can discover. So instead of everything being uncausal and just small islands of causality, he wants to inverse the process. You want the island in the sea metaphor inverted to be a notion of causality with just a few islands of unexplained system-wide behavior. Phenomenology will inevitably result from research focused exclusively on a very small fraction of the components of the system, their interaction. Thus, unrooted causality explanation invariably produces phenomenological information. In the 20th century biology, last century, there are many islands of causality floating in the vast sea of stem collection, phenomenology. System-wide rooted causality explanation inverts this relationship so that when successful, it gives rise to a framework of causality within which are always to be found islands of not as yet understood phenomenology. Axiom 5. High-resolution quantitative and qualitative observation are irreplaceable. Despite the fact that we want system-wide, we need observations at various levels. And finally, axiom 6. Explanation, mechanism can only be revealed by deliberate experimental perturbation and predictive challenges to the system. Experimental perturbation, cause and effect. Only if you perturb, you do an action to perturb and then measure after, only then you can talk about causality. So we have this philosophy of Eric Davidson with respect to defining causality in molecular biology. How am I doing this time? I'm okay?
Yeah.
A very important paper that I did with him was about the regulatory genome and the computer. So we did this together with one of his students, Ben De Leon, Tabu De Leon. So what we did here, we showed how the information system in the cell is presented by making a comparison component to the electronic computer. So there is a computer in the cell called the regulatory genome. and there is the electronic computer. How do you do comparison between the two? What is hardware? What is software? What is memory? What is fault tolerant? What are wires? How is the information transmitted? We did it one by one, and by doing this, we actually presented a homage to the 50th anniversary of the book by von Neumann, The Computer and the Brain, where he did exactly that, comparing every aspect of the brain as information processing and the electronic computer. Step by step, every component in the brain, in the the materials in the brain versus the hardwired electronics and things that are in electronic computer, step by step. But we did the same for the sea urchin embryo, and this was published at 50 years from von Neumann's seminal book on the brain. Here is how Boolean logic looks in the cell of the sea urchin, and in every cell. We have AND logic, OR logic, the NOT gate, and various compositions when we diagram them. Here are the truth tables, and here is how they are implemented in the cell. You see it with transcription factors and genes, and here is the biochemical diagram of how the biology operations are implemented. So it's a fascinating way to talk about principles in the cell. And there are many things to talk about it. I want to complete maybe the presentation by showing you the much advertised result that they were able to prove completeness of causality for the gene regulatory network. And this is the work that was done between Isabel Peters and Eric Davidson. Although many people contribute to the entire operation, including myself, part of the paper with Eric on logic functions of the cis-regulatory code. But the Boolean model that I'm going to introduce you is the most fascinating thing that they invented, and this was able to prove completeness of the 50 gene network called endomesopyle. So you see here, this is how we represent genes. There are genes and they go and bind somewhere. And there are regulatory regions of the gene where other transcription factors bind, and this gene is a transcription factor, goes and bind to other places as well. So this is the dynamic of what's going on. So let's see what we're doing from here. Here is how the network looks when it's measured in the sea urchin embryo with just a few cells at the beginning of life of the embryo. And fascinating topics to talk about in detail. This is the entire endomesoderm gene. 50 genes involved in the endomesoderm network, gene regulatory network. And they proved this thing complete. Imagine that, thinking big. So the Peter Davidson Boolean model. So they published in the Proceedings of the National Academy of Sciences this paper. Look at the title. Predictive computation of genomic logic processing functions in embryonic development. Predictive computational genomic logic processing function. So they also co-authored the last book that they published, which is Genomic Control Processes, Development and Evolution. So I'm going to get to the Boolean model. So you're going to see how thinking and how development is as a dynamic system. And we have the prediction aspects and how to integrate everything together. So the Boolean modeling of gene regulatory networks involves something called vector equations, which is a programming language that they invented that can be used to implement, to simulate the actions in the network by genes, the wet lab experiment. So there are GRN rules, and these Boolean equations are going to be used to implement the entire endomesoderm gene regulatory network as a set of Boolean equations, if you can imagine. Here is how a Boolean equation goes. You have a gene that is on or off. So you have this idea of a variable that you can turn it on and on. So you have now Booleanity at least defined. And here you have various of logic function that exist in the gene regulatory networks of sea urchin and logic or logic. You saw a number of them we discussed. And here is how vector equations work. One of the fascinating things, they discovered a time unit. So there is the time unit, which is for them 3 hours, which is fascinating. 3 hours is exactly the time when you need a new transcription factor. You go through the cell membrane to the genome, which magically opens exactly the gene you want. Nobody knows why. And then you copy that gene and you do it as an mRNA, and you take it back through the membrane of the cell in the cytoplasm, where it becomes the transcription factor as a protein. So this is exactly 3 hours till you get a new version of your transcription factor of interest. So they use this 3 hours as the time unit. And this program that they are going to write in network equation is a time-dependent process, so you need to ask at various times what you do. And here is how the equations look like. If at time 3, gene B is 1, and at time 3, gene C is 1, and it's not true that gene D is 1, at the previous unit at time 2, then else is 0. And all these if-then-else things, here are all the equations for the endomesoderm network. It's a mountain of equation programmed in this Boolean equation framework of there. And the amazing thing is the following. This is the genius and the asking big questions of Eric. So Eric said, all right, we take this gene regulatory network with 50 genes. Let's look at the most demanding experiment that was done on them. There is a one in which they duplicated the anus of the sea urchin, so it has 2 anusi, whatever. So it's an extremely complex process. And they did all the measurement. And this happened 10 years ago, but they have all the measurements. So Eric said, we're using the network equation to predict the values, and they better match the experimentally measured values in the cell. And here is the match. It's almost perfect. The yellow things are mismatches, but the overwhelming things is, look how many genes are listed. About overwhelming ones are predicted at the right time on and off by the network equations. Those are a few. It's biology, nothing is perfect, but it looks enormously completely predictable, almost completely predictable. And this is unbelievable to obtain the measurement values for these equations that simulate the network. Here is another example of a set of genes and where the errors are the red things and so on. And so on and so on. So in the conclusion, the Boolean model shows that the endomesoderm GRN contains sufficient regulatory information to recapitulate developmental gene expression. Almost complete experimental analysis of this early stage, the pregrastular gene regulatory network. The Boolean model demonstrates that integration of cis regulatory information produces the correct system-level output. The proof of the GRN concept that was put forward by Eric as the model to be inferred. How do you do with perturbations? There are experiments and everything is fine. So in the end, what you see in the cell, in this regulatory genome, the computer in the cell, you see this parallel. The electronic computer is made out of gates with everything 0 and 1. You connect to the next level of gates and you have this circuit. When you have an input, when you get 0, it's a Boolean circuit, it's synchronous. Everything works in a bit that is doing every operation synchronous. And everything is well defined, it's a completely defined gate. How is in the cell? It's a Boolean circuit. It's a little bit like the internet. It's asynchronous. When you want things to happen, sometimes it comes 0, sometimes it comes 1, sometimes 1.4 variations. When it connects to the next level, it recruits other things to come in, and the result is 1.1 or nearby. So it's asynchronous and incompletely defined gates, but it has this fault tolerance because the draconic synchrony here, the moment there is an error, needs to be stopped to find the error, you do much better in the electronic and the cell system there. So the three components of the computation in the electronic computer are the program, what you write, C, the Fortran, Python. Here is the logic workflow, and here is the hardware where the transistor is the workforce. If you look at the corresponding computation workflow in the cell, the software code is what is our DNA in the regulatory regions. The logic workflow is what Eric Davidson and his 300 students, through systematic perturbation, infer as the meaning of what's working, encoded the DNA. And at the hardware, what is the most interesting thing is that you put a piece of DNA and the cell has floating components that bind to it and form the protein machinery to work like a computer. And the transcription factor is the workhorse corresponding to the transistor. And it's a self-assembly computer. You put a piece of DNA, it self-assembles the hardware around it. You put a different piece, it self-assembles in different ways. So it's a totally different principle than the brutal order in the electronic computing. Everything needs to be perfect. Here, there is a lot of flexibility, for example, to evolve, to do better in computation. I wrote an essay about lessons I learned from Eric, and I would like to just end this talk by talking about my lessons. Axiom 1. Serious people do things seriously. He was the toughest scientist I ever had the pleasure to work with. Axiom 2. The answer to how to recognize the right place to crawl through the wall of thorn lie in the shape of the mental triangle composed of intuition, current knowledge, and logic. Axiom 3. Logic rules. He was the logician of molecular biology. Axiom 4. If we care about anything outside our current events, we need to care about those who will carry the brilliant torch forward. The students, we need to care about the future scientists. Axiom 5, life is short, let's keep in touch. Axiom 6, use it or lose it. He was a big proponent. And Axiom 7, the most beautiful of all, have inexhaustible optimism, inexhaustible curiosity, inexhaustible energy, and inexhaustible honesty. I typically end with the Beatles, Lennon, McCartney, and in the end, the love you take is equal to the love you make. So this is me at Caltech. Here is Eric, me, and my student Eric Ryan Torpine. Eric and me co-advised him. This is in the last month of his life in his office. Here is me and Isabel Peter, and here is me and Eric, surrounded by the beautiful sea. Thank you very much.
Thank you very much. We've got some time for questions if you'd like to follow up on the particular items. Yes, microphone. We get to use the mic.
Beautiful talk. I tried to understand most of it. I am not a computational biologist, but a biologist, so it was very fascinating to me. So my question is, when you talk about this Boolean circuit, and you talk about the ill-defined logic gates. So what is the role of transcription factors, disorders in transcription factors? Like, can we account for the disorder, or we know a lot of transcription factors regions are like intrinsically disordered or unfolded, like how they fold or how they interact with the DNA. Can they fill the gaps in the knowledge of?
Well, those are very regulated. The transcription, the protein structure are very well defined. Those are the workhorses in the organism. Every organism has more or less the same transcription factor families with fewer or more members. But those are very, as far as protein structure, are very regular. So the issue of recently more interesting, the topic that you're studying, the protein disorder, I am not aware of the connection with the regulatory genome. It's a much less studied phenomenon. I'm sure there should be interesting topics to such.
Like, there are some mRNA transcription factors like proteins like TR1 and HNRNP, if I'm not missing out on something, but those are like those form fuzzy complexes.
Yeah.
And they are disordered, they go liquid phase separation.
They say that the disordered part are because of protein-protein interaction. They need to form mutually complementary parts. So, but I don't know too much the connection with cis regulatory logic for that. So let's start.
Thank you so much.
Thank you.
Thank you very much for the presentation today. I just had out of curiosity, you talked about there being parallels in the sea urchins and the computer. You mentioned that there's junk DNA. So what's the junk DNA equivalent to the computer?
Very good question. We don't know. We looked at only known mechanisms that match the electronic computer and principles like fault tolerance and so. So I think my ending slide in which we have the self-assembly computer, you put a piece of DNA, the hardware is assembled, allows the computer to evolve, to get better, because biology can mutate DNA and other things can. So it may be that the junk DNA is a territory for innovation, for new proteins that can come in place and so on, which is not available to an electronic computer, but we can only speculate.
Thank you for a wonderful talk. So I have a question. So in your logicon and Boolean operators, you kind of assume that all the signals are binary, either 1 or 0. But in real life, proteins can be expressed at different level, and their function wouldn't be able to reach a threshold level if their expression was too weak. Have you ever seen that certain inputs are present, but the signal is too weak, not able to reach the truth table you have described?
Very interesting. So typically, all those things are collected in a lifetime of research by Eric Davidson. And I was the mathematician on board, the model and the C principles. And indeed, some of them are multi-input. And it still gets a certain truth table associated with it, but it's much more complicated. So for example, one of the deepest mysteries, we cannot compose them in a similar way in which we compose the electronic circuitry of the electronic computer, to make a hierarchical building. What you see 2 or 3 combined, so compositionality is a big mystery. How it builds deeper and more fault-tolerant circuits, but we couldn't find out the rule of composition.
So put it another way, your selected choose table are the ones that you really find them composable. But there are some other choices that you wouldn't be able to solution, and so you couldn't find them.
Absolutely, it's not an exhaustive repertoire. It's the one discovered so far. It shows the importance of thinking in a similar way in Boolean logic terms to understand function of living organisms. It's possible.
Thank you very much for this beautiful talk. You were talking about the so-called junk DNA. They are not really junk, right?
Of course.
Those non-coding DNA, they actually have many different functions.
Absolutely.
From your computational studies, can you predict those non-coding DNA functions? Because they can also code for RNA, which are not a message RNA, right? It could be microRNA and other RNA.
Well, 1/3 of the genes in the organism, we don't know what they do. So it's still a lot to discover. And people did synthetic organisms with minimal gene structure to try to understand those questions. So we're still along the way to try to find answers. So I don't know more than that.
You know, when we teach biochemistry, we sometimes also talk about the gene regulatory circuit. You know,
Yes.
like you have activators, suppressors,
Exactly.
all kind of gene regulations. Can you predict those regulatory functions in the computational models?
Some of them were cataloged, and our paper is actually a long list of logic functions discovered. And at least is conceptually to see the similarity with Boolean logic in these guys presented there, but still there is no escape in order to understand that Boolean logic is fundamental. But it appears in a very different setting that the clean mathematics of what we learn in engineering or computer science courses. It's a much more complex way, and we still miss the fundamental understanding of compositionality. How can you take two of those and try to build something new with it? It's very mysterious how things work. So a lot of mystery remains.
I have another question, if I may ask. We discussed before your talk about using AI as tools to understanding the functions of genes and proteins. What do you think about this AlphaFold? This.
Oh, I know very well. I work in this area.
So, do you think it can really replace X-ray crystals, structures,
So,
MNR and cryo-EM, or we still need the experimental data?
I think it will not get to cryo-EM immediately. Be a long way. I am a quite critical person with respect to AlphaFold because I work in this area for almost 30 years. But I work on rigorous methods, so AlphaFold and all this deep learning is like boxes and things not explained, and there are many things that are not completely honest in the presentation of the papers. But I can tell you that specialists trust AlphaFold, at least in a first order of magnitude of the structure involved. Of course, they don't fold everything. I think they fold well when there are many sequences in the multiple sequence alignment of the protein you predict. When there are very few, they are sketchy and they over beautify their result. They are not very good at new proteins and so on. But is there an achievement to be recognized as a breakthrough? I think so. but it can be done with more honesty and scientist to scientist dialogue as opposed to the, bravado, I work for Google, so I can afford to run all the we tried to run their program, nobody can. You need to run something of somebody else's. So they are secretive. They didn't participate in the last CASP, although they are the big experts. So there are many things
Oh, no.
a little bit less fully honest that I would prefer them. But overall is a big achievement in the sense that at least you have a solution that you think it's working, but you don't understand it. And still accepted by the community, which is ridiculous in
But
some.
not really accepted. People,
It's still controversial in some way,
yeah,
but
yeah. People still want to see experimental data.
yes.
Yeah.
But I saw predictions for very large proteins that they capture quite a good percentage in the right way. So I was impressed. So they do some things good, but maybe not. We solve the problem. It's
Okay,
somewhere along the way.
thank you very much.
My pleasure.
Any closing questions? And, great. Well, Dawn, did you want to have any closing remarks?
Thank you for being here with us and for being in the Pepper Center. Here's to have you. Such a wonderful talk.
Thank you for organizing all this and our discussion that I learned a lot about the university and about philosophy and it was a great visit and I thank you very much. You are the heart of organization. Thank you.
Thank you very much.